Hello everyone. Today we are going to talk on inoculum development. What is the need of inoculum development? This is carried out to reduce the lag phase during the fermentation process. Now, this is the purposely I have taken this picture. This is to realize that how big are fermentation tanks that we use at industry level. See, normally whenever we inoculate with the help of wire loop, that time we inoculate it in the flask, isn't it? So, inoculation with the help of wire loop is possible in case of flask, laboratory flask. But when we think about the industry level fermentation tanks, so such a big tanks, fermenters are available and we have to inoculate in this tank. Now you just imagine that if whether we are going to climb on a ladder and then inoculate in the ferment in this fermenters, no, this is not possible, isn't it? So for this purpose there has to be a particular process, and that is known as the inoculum development. Inoculum development is usually carried out in a stepwise manner. Now let's discuss the what are the criteria for inoculum development. The volume of inoculum should be such a large in size that it should help to minimize the lag phase of fermentation. Now what exactly happen in lag phase whenever we inoculate culture in a fermentation medium? That time this culture try to adjust itself into that production medium, isn't it? And that period is known as lag phase. So the inoculum development is carried out to minimize the lag phase so that overall fermentation period get reduced. That is what we want, because if the fermentation period is too big, it get extended. In that case there is a chance of contamination. And the very important is that whatever the culture which we are inoculating, which we are going to use, that culture should be metabolically active, okay, and it should be genetically stable. Otherwise, after the inoculum development, if we use this culture for the fermentation, then if that culture undergo any sort of mutation, then there is a possibility that this culture may lose its ability, its potential to produce the product, and therefore the inoculum culture should be genetically stable. And very important is that it should be free from contamination. Okay, next, whenever we carry out the inoculum development process, that time our aim is to increase the biomass. See, its objective is not to produce the product. That's why I've written that formation of product is not an objective during the inoculum development, and therefore the medium which we use for the inoculum development, that can be different than the medium which we use for the production, for the actual fermentation, because during fermentation the medium which we use, we call it as a production medium. The composition of the production medium is designed in a such way that it should support the product formation, whereas the medium which we use during the inoculum development, here the our aim objective is not to get the product. Here our objective is to cultivate the culture, is to increase the biomass. See, these are two different objectives. Again I'll repeat, during the fermentation process The production medium is designed in a such way that it should support the product formation, whereas the medium which we use during the inoculum development, it should support the mass cultivation, biomass cultivation. It should not design to support the product formation. Okay. Now, the lag phase in the production medium can be reduced by using the inoculum development medium, which is sufficiently similar to the production medium. Even if I mention that here the objectives of inoculum development is to support the biomass or the biomass cultivation, and the aim of production medium is to support the product formation, but still, even if the objectives are different and if we use the very different media for inoculum development, if there is a tremendous difference between the inoculum development and the development medium and the production medium, then again there is a possibility that the lag phase in the production medium may increase. So if you want to reduce the lag phase in the production medium, then the medium which we use for the inoculum development should be sufficiently similar to the production medium. Okay. Generally, the quantity of inoculum which is used, it's between 3 to 10% of the medium volume. If if your medium is 100 ml, then you should use the 3 to 10 ml of your inoculum, and like that way, as you keep on increasing the volume, medium volume, accordingly you should increase the inoculum size. Relatively large inoculum volume is used to minimize the length of lag phase, and thus it will help to generate the maximum biomass within a short period of time, and thus it will help to increase the product. Now, the inoculum is always built up in a number of stages. See, the inoculum development is never carried out in a single step. It is carried out in a number of stages, and which results in production of sufficient biomass, and that biomass is used to inoculate in the production medium. Now, this as I said that it is carried out in different stages. This stages may involve 2 to 3 stages in the shake flask and 1 to 3 stages in the fermenter. But if in that case also, if greater the number of stages in the inoculum development, again the time will be increased and greater will be the risk of contamination. So you have to keep the balance between the number of stages used for the inoculum development and the larger volume of inoculum. Okay. Now, these are the general steps for inoculum development. Number one is you take the master culture and reconstitute the master cultures, means try to revive that culture, and for that you spread it on a solid medium. Then after incubation of those plates, select the colonies, and again you inoculate it on another solid media that you consider as a submaster culture, and you have to use this submaster culture henceforth for every new production run. But before that, you must check the productivity of the selected culture. Check the productivity where, check the potential of this culture, whether it is really have that potential to produce the product in which we are interested. And once it is checked, then you inoculate it in a check flask uh, having a capacity of 250 to 500 ml, containing 50 to 100 ml of the medium. Then incubate this shake flask at either in a shaker or at room temperature as per the requirement. Then inoculate this inoculum into a larger flask. Again you incubate this flask and use this biomass to inoculate it into laboratory fermenter, incubate it, and then after incubation, whatever the biomass which is cultivated in the laboratory fermenter, use that biomass to inoculate it into pilot scale fermenter, and this is how the inoculum is developed in a stepwise manner. What is important in all this process is you have to check the culture purity at every steps. Now, suppose if there are many fermentation processes where the inoculum we use the fungi, that is the mycelial organism. Now we can, or there are some fermentation which uses fungi or actinomyces as a uh, producer strain, production strain, and these fungi or streptomyces are capable of producing the asexual spores, and so these spores are used for the inoculum development. Now, see, there are two ways for the inoculum development using the spores, or what we can say when the spores are used as a inoculum. Number one is there are some fungi which develop the spores on a solid medium, and here the roll bottle technique is used where the molten agar, the agar medium is poured in the bottle and it is rolled on the surface, so that that agar medium is set in a cylindrical shell inside the bottle, and simply it provide the large surface area, and then you inoculate it so that you will get the large surface area and the spores can be cultivated. And another way is by using the submerged culture. This is the very easy way to cultivate the spores on a large scale. So thus I have just mentioned two ways for the cultivation of spores. One is the use of a solid media where the large surface area is employed, and the second one is in the submerged culture. Okay. Now, this spores cultivated by this either of this technique can be used as a inoculum. And there are some fungi which do not produce the asexual spores. In this case, the vegetative mycelia are used as an inoculum, and therefore you have to develop or you have to design the medium which will support the growth of vegetative mycelia, and use that vegetative mycelia, consider it as a subculture, and use this subculture for every new fermentation batch. Then this is the first stage of seed culture. Second stage of seed culture, the objective of this seed culture is to just to increase the number of cells per ml, and finally seed culture is adjusted as per the requirement, means you prepare the seed culture in the volume which is required to inoculate it in the production medium. Okay, but in every step it is mandatory to check the culture purity, just to ensure that there is no contamination. Now this is the overall inoculum development program. See, this is the master culture, and from this you take a loop full of culture and inoculate it into 30 ml and incubate those flowers on rotary shaker for 5 days, and then use this uh, fully grown culture to inoculate it into second seed medium in 2 liter flask. Then incubate this flask for 2 days on a rotary shaker, and then again whatever the cultures which are grown in this second seed culture, collect it and inoculate it into 15 liter of a third seed culture medium, and incubate it for 2 days, and again inoculate this 15 liter of third seed culture into 60 liters of fourth seed medium, and then 60 liters of fourth seed medium is inoculated into the 1000 liter of fermentation tank, and this is how the inoculum is developed in a stepwise manner, and it is ensure that there is no any contamination, and the whenever that inoculum is inoculated in the production medium, lag phase is reduced. So